In this explainer, we're going to look at how precision nutrition is being applied to manage cardiometabolic diseases, things like cardiovascular disease and diabetes. You see, while we know that broad dietary patterns like DASH can be beneficial, when you dig into clinical trials on specific nutrients, the results are often all over the place. That inconsistency points to a fundamental issue. We all respond to food differently. And that's the core problem precision nutrition is trying to solve. So, here's our roadmap. We'll start with the limits of our generalized one size fits all advice. Then we'll get into the core concepts of genes and the microbiome, followed by the key mechanisms at play. We'll walk through a case study on glycemic response, tackle some common misconceptions, and then wrap up with a summary. The central question we're addressing is why an intervention that works perfectly well on average can completely fail or sometimes even cause harm for a specific individual. To get to the bottom of this individual variability, precision nutrition focuses on two main biological factors: our unique genetic makeup and the community of microbes living in our gut. So, how do we quantify that genetic risk? One of the main tools is the polygenic risk score, or PRS. Now, you've probably heard about single gene variants, like the APOE4 gene and its link to atherosclerosis. Those give us some information. But a PRS is much more comprehensive. It combines the small effects of many different genetic variants to create a single score. This gives us a much more accurate estimate of inherited risk, often improving our predictions beyond what traditional clinical factors can tell us. It's not just about our own human genes, though. The gut microbiome has a direct line to our cardiometabolic health. A classic example is the production of a compound called TMAO. Here's how it works. Certain gut bacteria metabolize compounds like choline and L-carnitine, which are found in high amounts in red meat. A byproduct of this process is TMAO. When levels of TMAO are high in the bloodstream, they're directly associated with an increased risk for thrombosis and atherosclerosis. It's a clear pathway from diet through the microbiome to disease. Okay, so we have these individual factors, genes and microbes. Now let's examine the actual mechanisms through which they influence disease, and we'll use type 2 diabetes as our focus. Our whole view of diabetes has changed pretty dramatically. We used to think of it almost exclusively as a disease of the pancreas, but now we understand it as a complex systemic disorder. It involves this intricate communication between the brain, the gut, muscle, and adipose tissue. And because our understanding has expanded, treatment strategies have to as well. It's no longer just about managing insulin, it's about addressing systemic problems like visceral fat and chronic inflammation. Epigenetics offers a key mechanism for how our environment interacts with our genes. This slide shows a perfect example. If the pancreatic islets are chronically exposed to high levels of palmitate, a very common saturated fatty acid, it triggers an epigenetic change. Specifically, it causes DNA methylation. Think of this as little chemical tags getting attached to the DNA that alter how genes are expressed, without changing the DNA sequence itself. And the functional result? A measurable drop in insulin secretion. It's a direct link from a dietary component to altered gene function. Of course, to make any of this practical, you have to be able to measure these individualized responses. This is why technology like continuous glucose monitoring, or CGM, becomes so important. It's truly foundational to precision nutrition research. By tracking glucose levels every few minutes, a CGM gives us a high-resolution movie of someone's personal metabolic response to specific foods. It lets us pinpoint exactly where and when an individual's metabolic traffic jams are happening. Now, let's look at how all these principles come together in a practical application. We're going to examine a landmark 2015 study from Zivi and his colleagues that focused on predicting these very individualized glycemic responses. So the study design was quite elegant. They had all the participants eat identical standardized meals. They then used CGMs to measure everyone's postprandial glucose response, or PPGR, which is that rise in blood sugar after you eat. At the same time, they were collecting a huge amount of clinical data and detailed gut microbiome profiles. All of this information, the food, the glucose response, the clinical data, the microbes, was then used to train a machine learning algorithm to predict how a specific person would respond to a specific food. And this chart really shows the headline finding of the study. It's stunning. You're looking at different people who ate the exact same meal, and their glucose responses were wildly different. Some people had a very low, gentle curve. Others had a huge spike. This is a perfect visualization of that inter-individual variability we've been talking about, the very thing that generalized advice just can't account for. The conclusion was pretty clear. The machine learning algorithm, which integrated all that personal, clinical, and microbiome data, predicted a person's glucose response far more accurately than the standard method of just counting carbohydrates. This was a powerful demonstration of the superiority of a person centric nutritional model over a purely food centric one. Now, whenever we discuss concepts like genetics and the microbiome, it's easy for oversimplifications to creep in. So let's take a moment to clarify a few common misconceptions about the roles of our genes and our early life environment in chronic disease. One of the most frequent misconceptions is that your genes are your destiny when it comes to diseases like type 2 diabetes. The data simply doesn't support this. While genetics absolutely play a role, the common variants we've identified so far only account for about 10 to 15% of the heritability. This tells us that the lion's share of the risk actually comes from environmental factors, and crucially, the interaction between those factors and your unique genetic background. And that window of environmental influence opens well before we're even born. According to what's known as the Barker hypothesis, metabolic conditions during pregnancy can essentially program a fetus for diseases that show up in adulthood. For example, if a mother experiences metabolic stress like high blood sugar, it can cause lasting epigenetic changes in the fetus that increase that person's long term risk for developing obesity and type 2 diabetes. All right, let's bring it all together. We'll quickly synthesize the key concepts from this explainer, and then I have a few questions to help check your understanding. So, the key takeaways are these. First, precision nutrition is an integrative science. It combines genomics, microbiome data, and information from wearable sensors. Second, its goal is to shift us away from generalized guidelines and toward individualized, data driven plans. Third, this approach makes it possible to predict personal metabolic responses to food. And finally, the future of this field likely involves AI driven systems that can provide us with continuous real time feedback to guide our behavior. Okay, first question for you. How does the gut microbiome contribute to atherosclerosis risk through the metabolism of dietary choline? Question number two. What was the primary conclusion of that 2015 YIDI study regarding postprandial glucose response? And the final question. Explain how exposure to palmitate can epigenetically modify the function of pancreatic islets to decrease insulin secretion. If you found this useful and want to see more explainers on complex topics like this one, please subscribe to the channel and give this a like. Thanks for watching.